Good afternoon and welcome to today's CottonInfo and Australian Cotton Disease Collaboration webinar. I'm Sharna Holman, CottonInfo's Technical Lead for Biosecurity and Disease, and I'll be facilitating today's webinar. Today's webinar is focused on sharing the latest update on how our industry is working towards minimising the impact of disease in cotton and some of the trials and research underway this season as part of the Australian Cotton Disease Collaboration. You'll be hearing from a range of presenters, including Susan Maas from CRDC, Dr. Sam Periyannan from UniSQ, and Dr. Linda Smith from Queensland DPI, as well as Bob Ford from CottonInfo. They'll be providing an overview of the ACDC program, key research areas which the program encompasses, and some of the trials that are underway this season to address some of the cotton industry's most challenging diseases.
Thanks, Sharna. To kick off today, I'm starting with the story of how we got to a pretty exciting initiative for CRDC. So as part of, our 5-yearly cycle of strategic planning, and as we were working towards the current strategic plan, which is Clever Cotton, it was really apparent that, there's a lot of concerns about disease. And that the issue was sort of getting worse and not better. And really, that, need for industry research to try and help with that. So we set a really clear goal in the strategic plan about reducing the economic impact of cotton diseases to less than 5% of cotton production. It's sort of, you set a big goal and then you go, okay, well, how are we going to get there? And the reflections we had was that, CRDC has had a long commitment to disease research. So it's not that commitment or investment in research was necessarily the problem, but it was really about why weren't we seeing that impact come through? So we called on Professor Mark Gibbard from the Centre for Crop Disease Management at Curtin University and just got a fresh set of eyes to sort of really, look at what are we doing in disease, how could we do it better, and, what might need to change. So there's lots of feedback from the review. I think it was a really good, fresh process to go to. And the review identified that we needed some structural changes to kind of really facilitate being more strategic and being more engaged. And thinking about yes, investment, but also thinking about how we can coordinate on disease issue and really, resulting in what is, hopefully that sort of strong national strategic leadership. So as through a process then, the concept of the Australian Cotton Disease Collaboration was launched. So for CRDC, that's a $13 million investment for this strategic plan. And I think importantly, we're now at a point where we've got our strategic partners, so UniSQ and Queensland Government on board. And obviously we're going to hear a lot more. I'm kind of conscious I don't want to steal anyone's thunder, but certainly we've got that sort of strategic collaboration on board and have been through a process of strategic planning. So reflecting, having been with CRDC for a while, we were doing lots of activity, but it wasn't necessarily structured. It wasn't necessarily strategic. So by kind of taking a strategic plan and really looking at what are all the different elements that need to be addressed for us to move forward on disease and making sure we're thinking about how things fit together and the order of things, having that strategic plan and building in some monitoring and evaluation into that so that we can check in along the way and make sure, yes, we're actually meeting the expectations. So that strategic plan now, the ACDC team have put that in place and we've got people and projects and systems in place. So we've really tried to take a very structured approach to disease now. And again, Linda and Sam will talk to, the project and people that are in place. And I think the other part that we've got the systems for ACDC in place, and we're now really starting to work towards that. How do we continue to really drive on collaboration? And we've got, there's lots of effort in disease in the industry, and we're really looking at how do we partner with the broader industry and really bring together that strong focus on collaboration to solve, what is a critical issue. So more probably in this diagram than what you really need. And really this is just, flagging that we've got a structured kind of management around the ACDC initiative. And so there's an executive committee, Sam's the director, and we've got a technical committee. So that's, brings together the research sort of technical focus to make sure we're really thinking about, are we doing the best science? And importantly, we've also got the industry advisory group. So that's about making sure we've got the best science, but also that we're actually answering the questions and meeting the needs of the industry. And so we've now got those in place. And importantly, to flag, we'll link into the TIMS committee because fungicide resistance management is something that we need to think about. So that's a little bit on structure. And I guess I don't want to take anyone's what they're going to talk about away, but essentially within ACDC, we've got 4 research areas. So we've got Domain A, which is about systems-based disease control, Domain B, which is understanding pathogen behaviour, Domain C, spatial data analytics and modelling, and Domain D, fungicides and plant defence mediators. And so we'll hear a lot more about that today. And then I guess to talk through, there's a lot of diseases. What are the diseases? So we've got tier 1 diseases. So these are the things that are getting a really big focus. And again, you'll hear a lot of about those today. So black root rot, Fusarium and Verticillium wilt, and alternaria leaf spot. So, really a big priority on those. Emerging diseases and other diseases are certainly within scope. And so that includes, seedling diseases, your recurring wilt, northern issues such as target spot, grey mould, and boll rot. So a bit of an overview. So I will throw back to you, Sharna.
Thanks, Susan. So next we'll hear from Dr. Linda Smith, Principal Plant Pathologist from QDPI, who will be talking us through Domain A and C and what research is happening this season. Thanks, Linda.
Thanks, Sharna. Thank you for the introduction, Susan, to ACDC and the domains. So I'm going to just talk about the Domain A and C. So I did have a slide also just outlining our mission and the domains that we are working with. And these are key areas of research addressing the 4 domains, as Susan mentioned, and the tier 1 and tier 2 diseases that she mentioned also. So I'm going to commence just going through the Domain A and C and the objectives that we have for each of these domains and the projects that we currently have commenced. So the first objective in Domain is looking at tactical disease management, including farming systems and impact on disease management. So the first project I'd like to talk about is the Irrigation Nutrition Management Project, with the aim to reduce cotton wilt disease. This is led by Associate Professor Joe Foley for the irrigation part, and for the nutrition part of this project, it's led by Linda Schakowsky with Queensland DPI, and we have our partners UniSQ, DPI and CottonInfo. So the aim of this work is we want to quantify the impact that varying irrigation methods as well as nutrition management, what this has on impact on disease incidence of both Verticillium and Fusarium wilt under the different environmental conditions. Because we do know from overseas the literature that irrigation management and nutrition is very impactful on disease incidence and severity. However, it's very anecdotal, the information and data that we have in Australian cotton. So we have a focus on getting the data to really understand what is the impact on the irrigation methods and nutrition in Australia on these diseases. We have a dedicated field site in North Star. We're very, very happy and very thankful to the Coltons for providing this, because it allows us to do really strategic applied research on farm. And so we're looking at both furrow and overhead irrigation management on Verticillium at the Coltons farm. And we also have a planned trial at a Fusarium site on the Darling Downs. And this is the farm where Fusarium was first detected in Australia. So these findings from this work, they obviously will go into grower management recommendations. We have started the trials last season. So we have undertaken some furrow irrigation measurements to understand water flow, irrigation management, but we've obviously got to do a lot more work to understand the impact and how to manage that to reduce the impact of disease. The second project in this objective is looking at providing diagnostic support, developing up diagnostics for a quick result for growers, and also cotton pathology collection. So this work is being led by Dr. Dinesh Kafle and Dr. Murray Sharman at Queensland DPI, based at Ecosciences Precinct in Dutton Park. So as I mentioned, they're developing molecular tools for rapid diagnostics. Murray has already developed some of these LAMP assays in our previous project, and you'll be able to see from the diagram on the bottom right, he's developed this molecular quick assay where you can basically just select a little piece of discoloured vascular tissue from Verticillium infected stem, and within 2 hours we have a result that will determine if that is a defoliating pathotype or a non-defoliating pathotype. So very quick assessment at a molecular level, very accurate and fast to deliver diagnostics. And he's working on all the key pathogens using the LAMP assay. Secondly, obviously we'll have a database of the prevalence and distribution of cotton diseases in Australia. So just building on our knowledge over the years of having diagnostics at DPI. And we're also collecting isolates of various cotton pathogens and contributing these to a centralised pathogen collection. So this is something that Sam will talk about as part of Domain B, but a very, very important part of the research, because this will be the first time we've had centralised pathogen collection, which will be able to be used for other ACDC projects as well as other researchers outside of ACDC. So a second objective, A2, is looking at improved management options, through understanding the etiology. Now, the etiology of a disease is understanding what are the pathogens involved, what is their life cycle, what are the management options. So understanding really all about that pathogen and how we might manage it. And one of the key things for a pathogen is obviously is the carryover of inoculum into the next season. And so we want to understand, well, how does cotton residues, what is the contribution of the cotton residues and other residues to inoculum carryover of key pathogens, and how might we manage that to reduce that carryover? So the first phase of this project, which is being led by Linda Schakowsky with Queensland DPI, is doing a literature search just to understand, well, what are the tools? Molecular tools are key, but also what are the other tools that we could use to measure inoculum load in crop residues? And we also have been talking to potential collaborators like SARDI to understand, well, what are their molecular capabilities to help us understand this inoculum load and carryover. The next phase of the project will be then demonstrating that linkage between inoculum, soil type, the environment, farming practice to yield loss. And what would be the recommendations for managing those residues so that we're reducing the inoculum load and the inoculum carryover, and we have some good recommendations so that when you do this, this and this, you're going to reduce inoculum carryover and have less disease going forward. And we're specifically going to focus on Verticillium, Fusarium and Utapella, but also that doesn't mean we won't also look at some other pathogens as well, but we just really want to initially focus on those three. A second project in Objective A2, also related to spore inoculum, is just continuing some work that we've been doing with spore trapping to detect aerial spores of cotton pathogens. And this is led by Dr. Dinesh Kafle with Queensland DPI. We started this work with SARDI a couple of years ago because we wanted to know, well, Utapella species are supposed to produce aerial spores, and that is an infective propagule for Utapella species, which is causing reoccurring wilt. We have been working with SARDI to try and detect these. To date, we haven't. But we have detected other aerial spores, other pathogens such as leaf spot pathogens. So we want to continue using this methodology to look at the distribution. Well, can we detect Utapella? Is it actually out there? But also leaf spot pathogens. So we currently have some traps located in central Queensland for Utapella and northern Queensland with Samuel Kreckler for the leaf spot pathogens. And this will help us understand the etiology of these pathogens and help direct research to investigate management. For example, if we can understand the pathogen life cycle fully, we can then look at intervention into that life cycle to reduce the viability or incidence of disease. But also fungicide application, particularly for the leaf spot pathogens, because if we don't have an understanding of that dispersal of the spores and when disease takes place following that dispersal, we might be just randomly applying fungicides not in a strategic manner to control that disease. And that also raises questions about potential resistance development to the fungicides. So I think this spore trapping is really going to provide some insight into the life cycle and management of these diseases. Then our third objective within Domain A is going to be offering training and support for Northern Australia. Because we have cotton developing into the northern parts of Australia, we want to be able to provide pathology support where it is required, and also engage with obviously both states and collaborate with them on all ventures related to pathology. So the principal investigator of this project is Associate Professor Sam Periyannan at UniSQ, and all the partners, DPI, UniSQ, Northern Territory DAF, WA DPIRD and CottonInfo. So we want to understand what is the disease pressure in these regions and what are the key and emerging pathogens, because currently we actually don't know, particularly in Western Australia, what are the pathogens and diseases present. We certainly know that there are some emerging issues such as ramularia and charcoal rot, and so we will have a focus on those, particularly in the Northern Territory. And as part of this work, we want to build capacity through establishment of a diagnostic service, particularly because there is not one currently in WA. So we have been working with the pathologists there for developing that service, and also training in disease surveillance and also pathology training. This project proposal is not yet finalised, but we have actually commenced work. We've made a couple of visits up to Katherine and Kununurra, engaged in delivering workshops to introduce ACDC and how we can work together, meet the growers and the agronomists and the pathologists. We have done some diagnostic lamp training at Kununurra, and I've been up there late season this year to run through how you would do a late season survey and some of the diagnostics. And then we'll be going back up there for the early season to run through again the methodologies, just to make sure that we're all working on the same page to undertake surveillance diagnostics and deliver to growers. Now, I'd just like to now talk about Domain C, Spatial Data Analytics and Advanced Modelling. So this is a really exciting new area. We have a PhD student, Kingston Ocker, who is taking on this work, and the lead supervisor is Professor Ravnish Deo from the University of Southern Queensland. Sam is also involved, as well as Professor Adam Sparks from Curtin University in WA and myself. And the aim of this work is to model disease dynamics across regions by integrating historical disease incidence data, and that is the 20 to 30 years of disease survey data that we have been gathering from grower fields for many years. So we are going to take, I should say, Kingston's going to take that data. He's going to look at climatic and environmental variables, remote sensing, terrain attributes. He's going to turn this raw data into something that's meaningful and usable for industry. So the key outcomes, we're going to have an enhanced predictive and visualisation capability for disease incidence, improved understanding of climate and environmental drivers, practical tools for cotton growers to mitigate crop losses, and a broader contribution to agricultural systems with this modelling. So it's a very exciting space to be able to use this data that's been collected by Stephen Allen and Karen Kirkby and the Queensland staff. So all over the years, we're going to be using this data to model and to help predict disease. And there, a second project is being led by Associate Professor Alison McCarthy. This is also about spatial data analytics and advanced modelling. So in this project, she's developing a machine vision system to detect and differentiate between Verticillium and Fusarium wilts. So using this machine vision sensing to differentiate, she is using existing field trials. So using the trial at North Star, where we have the irrigation and nutrition trial, she is using remote sensing to gather data to differentiate between the two pathogens. So she's gathering data for Verticillium at Gedda, but she will also be screening other sites for Fusarium and get a signature for those pathogens and then be able to discriminate them early in the season before you actually see visual symptoms. And this then will be able to map fields with a high prediction of disease. In conjunction with this work, she is supervising a PhD student, Niraj Sharma. And currently they are about to start using this. Within this photo, you will see this amazing glasshouse with all this technology at UniSQ, be able to use all these sensors, multispectral indices, 3D plant mapping to look at the Fusarium and Verticillium in the glasshouse under controlled environmental conditions to understand how to differentiate between those two pathogens. So very, very exciting. That's a summary of the current projects that we have. And now I'm just going to, on my last two slides, it's just letting you know about the facilities and the research that's going to be conducted in New South Wales. So we have been working with the University of Sydney, the Plant Breeding Institute, to deliver a program for diagnostic capacity to New South Wales cotton growers. So this is going to be led by Dr. Tim Weaver, and they're so based at PBI. We will have a cotton pathologist and PhD students. They are going to also be running the cotton production course there now, and so there's going to be a building capacity for cotton at the Sydney University in pathology. So the outcomes of this collaboration is going to be that New South Wales cotton growers are going to be informed of shifts in disease pathogenicity that have agronomic significance, but also importantly, they're going to have access to disease diagnostics for the identification of important and emerging diseases, and also a decision support and management strategy along with that diagnostics. So we're very, very excited to be working with them to deliver this. And secondly, also down in southern New South Wales, a very, very important disease, as you all know, black root rot. And we're currently working with Charles Sturt University and Summit Ag to develop a project and to contract for them to deliver research on black root rot. This project is going to be led by Dr. Ben Stoddart, but it's also with Summit Ag, who are going to be providing the field-based science capability in field in that region. So the outcomes of this project is going to be improved understanding of black root rot disease incidence, cotton pathology support to southern New South Wales cotton growers, management options through effective farm practices. So there will be a lot of research in field on farm looking at crop rotation, nutrition, use of growth regulators and also biocontrol agents. And also through the course, if there are other options that look plausible for management, then they can be investigated. And also capacity building through co-supervision of a PhD student or students if it's applicable. So very, very happy, very pleased to be working with Charles Sturt Uni and Summit Ag. And I'm going to finish there and thank you for your time.
Thanks, Linda, for those insights into Domain A and C. So our next presenter is Associate Professor Dr. Sam Periyannan from UniSQ, who will talk us through domain B and D and what research is happening this season. Thanks, Sam.
Thanks, Sharna. Okay, I think Linda has beautifully presented the projects in A and C, and now I'm going to present a list of projects that we are going to do under domain B and D, and that is led by UniSQ. So as Linda pointed out earlier, one of the key objectives in domain B is to establish an Australian cotton pathogen collection. So right now, if you see, we don't have a centralized location where we can go and get any pathogen. So for example, pathogens in Queensland are collected in Queensland Herbarium. New South Wales are deposited in New South Wales Pathogen Curation Centre in Orange, and NT and WA haven't started yet. So one of the key objectives within domain B, and that is led by Dr. Cassie Percy, Senior Research Fellow at UniSQ, and in partnership with Queensland DPI, NT DAF, WA DPID, and CottonInfo, Cassie will be initiating a centralized and well-catalogued collection center for cotton pathogens that has been identified in Australia. So why we need this collection? This particular collection is really important for us to understand how does the pathogen being distributed across the different cotton growing belts and how they are evolving to overcome new varieties. And then this particular collection will also enable us to validate some of the isolate specific diagnostic markers that Linda pointed out. So only when we were able to screen a multiple isolates, we know that whether these markers are specific to particular strains. And the very important application of this cotton pathogen collection is to assist cotton breeders like CSIRO colleagues to screen their breeding lines so that we will know that whether the resistant lines that has been developed at CSIRO will have a broad spectrum resistance against multiple isolates, because they need to have this information if they want to release varieties for different cotton growing belts. And then coming to the second project, which is on host range and cotton differential set identification, which is also led by Cassie Percy. And as you all know that in Australia, cotton is often rotated with winter and summer crops. But the pathogen that are going to infect, it's not only going to infect cotton, but it's also going to survive in other host plants. So for example, in the previous investment, Linda has already identified host list for Verticillium. However, in this initiative, we are going to look at host for Berkelomyces roxia, black root rot, which is one of the really serious pathogen in the south. And we are going to look at the host range for Utipilla and also looking at host range for the newly emerging disease Macrophomina, which causes charcoal rot disease. And the other interesting thing in this project is we are also going to test the asymptomatic host. So if the plant doesn't show any symptom, it's not necessary that the plant won't carry any pathogen. There are a lot of plants which can act as a carryover for inoculum. For example, between the two cotton season, should be some living system where the pathogen can survive. So what we are going to do is we are going to test some of the asymptomatic host plants to see whether they still carry on the pathogen inoculum and build up for the next cotton season, or they are going to just minimize the inoculum load. And the third important point in this project is to identify cotton differential sets mainly to phenotype and group different Fusarium and Verticillium isolates. And the other important project within domain B is monitoring pathogen diversity, which is led by Dr. Alex Sotiropoulos, who has a lot of expertise on pathogen genomics. So we will be mainly focusing on studying the pathogen population in Fusarium and Verticillium and on other pathogens. Why we want to study the population? So we really want to study this in order to see how does the population structure in response to forming system or weather. For example, if there is a change in the variety, is there any population building up to evolve virulence to other particular variety? Or if there is a frequent change in weather, for example, frequent rainfall, there is a group of isolates dominating than the other ones to survive in the high humid condition? So those are the things we are going to, for example, identifying the factors that can influence evolution of virulence. How does the pathogen population develop virulence to overcome any new situations or environment or varieties? And the fourth important one, this is particularly important for some of the diseases that we are going to use more fungicides. Does the population is developing resistance to overcome the frequently used fungicides? And then the other important project within domain B is to generate reference genome for emerging pathogens, which is also led by Dr. Alex Sotiropoulos. For example, black root rot, Berkelomyces, and Corynespora, we don't have a genome reference, so we will be developing this genome reference. And this particular genome reference is important, for example, We want to know whether any new pathogen that is appearing in Australia, is it related to something from the overseas, or is it jumping from a natural vegetation into a cultivated crop? And the third important point is to develop diagnostic markers so that we can rapidly do surveillance. And the fourth important point is we need to understand how does the pathogen can evolve virulence, and also to see if there is evolution for fungicide resistant loci. And the other important project within domain B is to develop co-infection assays to study synergistic and antagonistic interactions, which is led by Dr. Sadek Balto. Sadek has a lot of expertise in the molecular biology aspects. So this particular project is very important because in Australia, for example, we have boll rot. So boll rot is something not only caused by one particular pathogen, it is caused by 3 different pathogens, for example, Coltotrichum, Fusarium, and Phytophthora. And there is also a chance that root rot will be caused by a different complex pathogen. For example, black root rot, dry root rot, and Fusarium, they can all together act together to form root rot complex. So one of the first objective within this project is to monitor if there is pathogen co-infection happening in the cotton settings in Australia. And then in order to identify factors, for example, soil type, rainfall, humidity, all these factors influence these pathogens to coexist. And the other important thing we want to study is the one particular pathogen is dominating over the other. Does all the presence of 3 pathogens are important to cause a disease outcomes? So there are a lot of activities that Sadek is going to untie from this particular project. And most of the work will be done in the glasshouse and laboratory settings. And then we'll be taking in the next phase into the field validation and field identification. So over the domain B projects, we are also building capacity. As pointed out by Linda, most of the projects includes PhDs training. For example, we have 4 PhD students for 4 different project activities. For example, Muhammad Zagib will be working with the team both in UniSQ as well as Queensland DPI. He will be going to characterize Utipila and identify the host range. And then Muhammad Amir, who will be placed mainly at New South Wales, Narrabri. He'll be working to understand how does the inoculum carryover happens in crop rotation. And then Amna Arij will be working with Alex and mainly studying the population diversity of Fusarium oxysporum. Then Muhammad Amir will be working with Sadek, and he will be going to standardize some of the assays, and he also going to study about the synergistic and antagonistic interaction between cotton pathogens. Then coming to the domain D projects, which is led by Professor Leventekis. So one of the key projects within this domain is the fungicide and plant defense mediators for Australian cotton industry inferred from national and global analysis. So what we are doing is, since we don't have much information about what are the different fungicides that has been used against cotton in Australia and overseas, this particular project will identify what are the different fungicides has been used and what are the different new products that has been in market that can be tested for future use in fungicide industry here. Thank you, Sharna.
Perfect. Thanks so much, Sam. We'll now hear from Bob Ford, Regional Extension Officer in the Namoi, who will discuss CottonInfo and the ACDC collaborations and involvement in local field trials. Thanks, Bob.
Thanks, Sharna. I guess from a CottonInfo point of view, we're looking at some of the actual diseases that are obviously cause as grief and have for some time over a period of time. And Verticillium wilt is one of them that we've, particularly down here in the Namoi, where I'm based, has reared its head for many, many years. And sort of understanding the disease matrix is very, very difficult because, we've seen now that there is various different strains that occur and they behave slightly differently within a crop, and management definitely impacts on how this disease behaves. So, but there is ways of helping with the disease, and temperature obviously is dependent with this disease. So in warm seasons, we don't see the actual virulence of disease as being bad, but certainly in cool seasons, we definitely do. So as an example, a farming systems approach combines knowledge of researchers, farmers, consultants, agronomists to come up with solutions. And I think we're all part of it in terms of what we're trying to do with this disease and come up with some approaches that will actually help. So I just want to talk about the QR code that you see there, and anyone that's online at the moment, they can scan that QR code. We're using an actual program called Group Map, which is basically a communications and research priorities tool that people can send information back, questions, ideas back, and it comes directly back to CottonInfo, which can be shared with researchers alike. And it's a pretty simple tool. You just scan it. You can use your phone if you want, and just once you put your email in, you can just go in there and push the plus sign and add whatever ideas you've got in relation to disease or questions. So we'd like everyone to actually do that if possible. But overall, we're trying to develop trials that may help farmers reduce the impact of the disease. And certainly crop rotation trials, as the pathologists have shown, there is an understanding of those non-hosts with, for instance, corn and sorghum interactions. And we're setting up a number of trials where we're trying to develop some management options using those crops, but also a couple of other crops, particularly butterfly pea, to get an understanding of exactly what's going on under that crop from a microbiology point of view. And it's hoped that obviously we come up with a list of management guidelines for not only those crops, but also some new ones that once we get a better understanding of what the disease is doing under those crops. Temperature control trials, we know that temperature definitely impacts on the disease. So, for instance, we've got a couple of trials there where we're actually looking at increasing the bed height and recording the temperatures of those high beds versus, beds that have slumped, etc. And we really want to look at actually when that disease gets into the crop. If those lower beds are actually contracting the disease, or the virulence is higher in something that's a lot lower and a lot cooler compared to something that's higher. We do know that, if you get the disease early in the season, typically you're when it really turns bad in sort of late January into February, that the case is normally worse. So you want to basically not have the disease getting into the crop in the first part of the season. And similar to irrigation, which was mentioned before, certainly when you're applying water or when you're actually planting, can actually instigate the actual disease depending on how cool it is. And similar sort of situation, if it's actually gets into the crop very early, you're going to have probably a lot of problems later on. So we've actually got a number of trials there where we're actually looking at specifically when you're actually applying water and when you're planting into, with temperature control systems. We've got plant physiology trials where we're looking at maturity, and it probably goes back to Steve Allen's work where, earliness is godliness. We're looking at trying to actually develop a compact plant type that's probably a little bit earlier than what we're seeing. I guess in some ways we've been our worst enemy as we've seen yields increase over time, trying to get that 15 bales per hectare sort of mark every year has been pretty consistent with many farmers, but there is a tendency to grow those crops out for a bit longer, and what that does is open those crops up to the actual back end of the disease, which can cause issues to that crop, but also can add a lot more of the actual disease leaf material going back into the soil. So in that trial there on maturity, we're trying to actually develop a crop that's yielding very, very similar to the later crops, but is just more compact, and we'll use growth control to actually do that. Skip row trials under overhead irrigation. A number of growers have actually said using 80-inch cotton that they've actually seen a lot less disease in those crops, and it probably makes sense that obviously they're not watering those crops as much, so they're not actually, the beds aren't as wet, etc. Also, there's a lot more air getting into the temperature, and the actual soils are probably slightly warm, obviously. But we can actually record the actual temperatures of those sites and get a good understanding, and we'll actually do yield and also do disease screening on those sites to see exactly what's going on. We also have a number of product trials which are with commercial products that are out there at the moment, just to get an understanding of whether we can see improvements. 0x has been around for a while, but we're we're looking at the microsclerotia really of what's going back into the soil, and also looking at the rates that are being applied using that product to see whether we can see any good results coming out of that, as well as some soil ameliorants and a disease trial where we're using a number of different products. They can be from a incorporation point of view, seed treatment, as well as actually foliar application, to try and see whether there's any of those products that actually help with the actual disease throughout the season. But I guess overall, we really want anyone out there that has any ideas or past practices that you believe have had an impact on the disease to please let us know. We know that a lot of growers and consultants do their own trials, demonstration trials out there, and we want to know about that stuff so that we can actually relay that back to researchers, and anything that actually farmers or consultants see that is worthwhile. It's worthwhile passing on to us, and that can be done through that QR code or ringing your local REO extension officer. And that's all I've got to say, Sharna.
Thanks so much, Bob. The group map is a fantastic avenue and way of getting growers and consultants to provide feedback and suggestions that we can definitely facilitate to the ACDC researchers and really get that feedback loop going. So it's a fantastic idea and concept. So that concludes the formal presentations for today's webinar. So thank you very much to Susan, Linda, Sam and Bob for sharing their insights and updates on the ACDC program and current research projects. And thank you to everyone who's been able to join us for this session. We really appreciate you taking time out of your busy schedule. And we hope you found this information really useful and helping highlight some of the disease projects that are underway. If you'd like to learn more about how to get involved with any of the projects, please reach out to myself or your local CottonInfo REO. And we look forward to seeing you at another CottonInfo event soon. Thanks, everyone.